want to tell you about an experiment that I do a lot, but I haven't really told you much about before, and it's a radiometric kinase assay with scintillation counting. Yeah, so that sounds like a lot of mumbo jumbo, but basically it's a method that we can use to measure the activity of kinases, which we talked about before. They're those proteins that add like negatively charged phosphoryl groups to other like proteins or other molecules, but I'm talking mainly about like protein kinases that phosphorylate proteins. And then they have this bulky group, but then the protein or the thing that the protein that they phosphorylate is like, what the heck, you just added this big bulky charged thingy on me, and then the protein goes, woo, or it moves some sort of way, and it changes in some form, and it changes the activity. And so the kinases are the things that actually put on those phosphate groups. And so if we want to measure that activity, one way, a really sensitive, like gold standard way or whatever, is with this radio labeled ATP. So radio labeling, we talked about that last week. We talked about radioactivity, where it's like when an atom has like too many nuclei, and like then its nucleus, so like the center of the atom part is like, what the heck, like this is really uncomfy, and then it lets off this radiation to try to like make itself more comfy. So yeah, more on radioactivity in last week's post, but the key thing is that you can detect this radioactivity. So I told you in the past about how we can detect it with things like autoradiography, where we put like a screen on top, and then the screen like captures the like the energy in like this metastable state, and then you shine light on it in this like scanner, and then it lets off light. Okay, well, we're not going to use that for this. For this, we're going to use the scintillation counter, which is this really old machine that I'll show you more later, but it's like seriously like this super like old school counter. And it has like a dot matrix printout. It's pretty cool. But anyway, so how it works is basically that we use ATP, that the last phosphate group, so ATP, adenosine triphosphate, so 3 phosphate groups, the last one, the gamma one, is the one that's going to get transferred. And we use ATP, that's gamma P32. So like that last phosphate, the one that gets transferred, is radioactive. So then you add the kinase with the hot ATP, so the hot is hot, just like slang for radioactive. So you have like, and cold ATP, because you don't want it to be like super duper hot. Like if it was all hot, it would be like, wah! Yeah, you don't want that. So it's just a little hot. But anyway, you add this ATP mix to the kinase and the protein, or the peptide. So a peptide would be just like the part of the protein that normally gets phosphorylated by this kinase. You can just like use the peptide part sometimes. More about that later. But anyway, so you do your kinase reaction, and then like you stop it. You quench it with like, so you add like EDTA to steal the kinase's metal, and then you add a ton of like a ton of cold ATP, so that any ATP, like any phosphorylation that occurs after that point, is going to be cold, and it's not going to like affect the results. And then you take that reaction and you put a little bit of it, you spot it. You spot it on these little filter papers. So if you look at like past methods papers or whatever, they call it like P81 paper, because it used to be like Waltman P81 paper, and then they discontinued it, and then the whole kinase community was like, ah! And then the Oak Hill lab started making it, and we're like, yay! But maybe so they make this like alternative paper, and so it like comes, you can like order like sheets of it, so I'll give you the link. And so we're really grateful that he makes it for us, but maybe we can make like this kinase assays go viral and get Waltman to make P81 again. Just saying. Anyway, but yeah, so thank you to the Oak Hill lab for keeping kinase alive. Okay, but anyway, so you can use like pins or whatever. I just use like needles because that's what I have, and this is a really bad example because I'm showing with an arrow. But so basically what I do is I cut up this paper into these like little rectangles, like these 2 by 1 centimeter squares, which if you don't have like a ruler like this, super duper handy, because it has like the edge or whatever, like you only have to measure from one side kind of. It's really, really handy. So yeah, I highly recommend one of these T thingy rulers. But anyway, so, and then I cut them out and I put them on, like, so this is just the top of like a styrofoam top from like a cuvette lid thingamabob. I steal them when they come in. And so I line them up and I stab So I do all this before, so I set this all up ahead of time. So I stab them and I like push this to the top and then put it in here because you don't want the paper to be sitting on or dripping onto the styrofoam. So then I line them all up in like pretty lines like that. I don't know if you can see that because I'm trying to show you my screen. Wrong place. There we go. You spot them like that. And you also spot like just the ATP mix, and you're not going to wash that one. Don't wash that one because that's going to wash off the ATP, which is the point of the wash step. So basically, after we do these, we put these filter papers, we're going to wash them and wash them and wash them. So we wash them in like a phosphoric acid, which I'll tell you more about later, why and stuff. But basically the special thing about this paper is it's phosphocellulose paper. So phospho, like the phosphate groups we talked about, they're all like negatively charged. And so you have negatively charged group on this paper, and you have negatively charged groups in the ATP. And so that'll keep the ATP from, the ATP won't bind the paper because they're both negative, 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 boop. Yeah, opposites repel, remember. Or like repel, opposites attract. So the negative charges are going to repel each other, and then it won't stick to the paper. So that's how you can wash off all that free ATP, because you don't want like the background, like all the ATP that didn't get added to your protein. You want to make sure like only your protein or your peptide is stuck on here, so only the like the hot stuff that's actually on the protein or the peptide is going to be on this paper. And then you stick them in these little vials, which are filled with scintillant, which is like this liquid stuff. So there's different kinds. This is the one I use. I put like 2 mils of it in here, so I do that ahead of time as well. And so usually it's kind of like viscous and gross and greasy. I use like a 10 mil like pipette or whatever. Suck it up, then do like 2 mils per one. Efficiency. But yeah, so anyway, I prepare those ahead of time, and then I take all of those in my little like my Santa sack that I take use to carry everything down across campus. And so I put the after I do the washes, so I do the washes in the phosphoric acid, and then I do a wash in acetone. So acetone, like if you've been to like a nail polish salon or whatever, and like you see when you put like the nail polish remover on or whatever, and then you like your fingers get all dry. Yeah, so the acetone's gonna like help dry the paper. So we like air dry it, and then we stick it in the scintillation vials, and then we stick it in that little counter thingy bob, and it goes. So they're in these like long racks, and then the machine takes them and it goes, and it goes a tube, and then a tube. And tube, and each time what it's doing is it's counting the light that's given off thanks to the scintillant. So basically in the scintillant is like this in addition to like solvent things to help like dissolve the stuff that you like the paper, then the like substrate, like the stuff you put in the tube. There's also like a fluor, so like a molecule that can fluoresce. So like a fluorescence is when a molecule absorbs light of one wavelength and lets off light of a different wavelength, or it absorbs energy of one wavelength and lets off light of a different wavelength. And so it's going to absorb the energy given off by the radiation, and then it's going to give off light, and then this scintillation counter can like multiply and detect that and stuff, and it's like super sensitive, and it's going to give you like these numbers. And then you have that like ATP only sample. Remember, you didn't wash that. You don't want to wash that, but you want to wash all the others. And that will tell you, then you can like compare. So that's ATP only is going to give you a readout of like how much signal corresponds to how much hot ATP. And then you know how much substrate and stuff you had. So substrate is the thing that the kinase puts the phosphate on. And then you can then like calculate based on that. Okay, so that was like this whirlwind overview. And now let's go like more in depth and like slow down. And sorry, I just kind of like whizzed through all of this, but I have like this whole like slideshow thing prepared for you now. And so I probably should have just like sliced this part piece by piece into the slideshow, but I made the slideshow before and I made this now, and now I got to get back to work. But hope you guys find this helpful. Oh, and be sure you label the paper in pencil. Pen will get solubilized or whatever by the solvent, and it's just gonna like come off, and then you like have no idea which piece is which. So yeah, so I make, when I do my little squares, I make them like 2 by 1 centimeter, and then like the top half centimeter, I make a line, so and then above that I label it with like A1, A2, or whatever, because the like based on like the scintillation racks, and then I put them in. And it's like the same, the order that I put them in my samples or whatever. But anyway, just label it somehow that you know what it is in pencil, because if you label it in pen, it's useless. Protein kinases are proteins that add phosphates, well, technically phosphoryl groups, to proteins. You can think of them kind of like as pencils putting on this negatively charged phosphoryl group. So it's like this phosphorus surrounded by oxygen. It's got these negative charges and it's all bulky. So it can cause the protein to rearrange a little and to regulate its activity. So maybe like turn a protein like on or off, kind of. They can also create new binding sites for other molecules now that you have this new charge group there. But then phosphatases remove them, resetting things. And then the kinase can come back and do it again. So this allows you to this regulatory system that allows for proteins to be regulated in terms of their activity and what they bind to. And it's also really important for passing signals. There's often signaling cascades where one kinase that's embedded in the cell surface, it gets phosphorylated, and then that activates it, so it phosphorylates another kinase, which phosphorylates another kinase, and you get these signaling cascades, and then each kinase can be phosphorylating more things, ultimately sending signals and doing things like altering what genes are expressed when, so what proteins are made when and stuff. So it's really cool. And but it can also be bad because if kinases go rogue or whatever, that can be associated with diseases like cancer. So there's also a lot of activity in trying to study selective kinase inhibitors, so drugs that can prevent these kinases from working. So there's a lot of reasons to study kinases and their activity, and how they can, whether inhibitors actually do inhibit their activity, so do prevent the kinase from phosphorylating stuff. So we need ways to measure this activity. One of the ways is with a radiometric filter binding paper kinase assay. I'm not sure what the technical term for it, but I'll show you what I mean. So basically, the idea is that we use radio-labeled ATP so that we can detect the transfer. So we talked about radioactivity last week, and that's basically, I'll show you more in the next slide, but it's a way that we can label that phosphorus, so then it'll give off this radiation, so these waves of energy that we can then detect. And so if we label the end phosphate, that gamma phosphate, then that's the one that gets transferred to the protein, and now your protein is radioactive and you can measure it. And so we talked about radioactivity last week, so if you want more details, go review that talk. But basically, atoms, so individual phosphates and oxygens, phosphorus and oxygens, carbons, hydrogens, all of those. They're made up of smaller parts called subatomic particles, and there's protons and neutrons and electrons. And basically, in a radioactive isotope, what you have is a version of an atom that has too many nucleus, an imbalance of neutrons and protons, so it's kind of uncomfy, and then it rearranges that nucleus, so that core where the protons and the neutrons hang out, and then it lets off some energy and particles and stuff as radiation that we can then detect, that released high energy radiation. So that's the idea. Again, lots more last week, but for now, just know that we have this radioactive phosphorus at the end, in that end phosphate group. That's the one that gets transferred to the substrate, so the thing that the kinase is actually adding to. So an overview of how we do it: we take our substrate. So like I said, a substrate is just the name for the protein or the peptide or whatever that the kinase is actually going to phosphorylate. So the phosphorylation is taking place usually in the context of a protein, in its natural, phosphorylation normally happens naturally. It just happens it happens within a protein or a peptide or within a protein. But we can also, to make these experiments easier, use just a peptide, so just a piece of the protein, just the piece that the kinase acts on. So kinases often have a preferred recognition or consensus sequence that they a site where they like that order of protein letters, and so they phosphorylate at a specific site on the protein. And these are often in like disordered or like linker regions, like linear type regions of a protein, not the really structured parts. So you can get away with just using that short peptide. But there's also peptides. Don't always work. I mean, like the kinase also can get, if it's like phosphorylating a more structured part, or if it's interacting with the protein in other ways or getting other cues. So the peptides won't work in all cases, but you can use this assay with proteins or with peptides. So I've actually worked with the whole protein, but you need to make sure it's really pure. So you don't like detect any background and stuff, and you have to take a lot of controls. But anyway, back to the story. So we have this, your substrate, so your protein or your peptide, and then you're going to add the kinase and the radio labeled ATP, so that we're actually like transferring something we can detect, because we can't detect just like the normal ATP. Then we allow it to phosphorylate, so we incubate it at a desired temperature, and so I usually use like 37 degrees, which is like normal bodily temperature, but you can also do different things. It depends on your kinase, but you just want to make sure you use the same temperature and keep it constant with like a heat block or something. Then after the set amount of time, which as we'll talk about, depends on whether you're trying to get like initial activity or like stoichiometry, so how many sites there are. Then you're going to quench it. So you're going to stop that reaction. And so now we have our substrate has been labeled with this radioactive ATP if the kinase phosphorylated it, but we need a way to detect it. And we also need a way to get rid of all the ATP that wasn't transferred. So like we have a bunch of the radio labeled ATP that's hanging out there with the protein and the kinase, and we don't want to detect that when we just want the protein. So we need to trap the protein and get rid of the free ATP. So what we do is we spot it on these little filter papers. So these papers are like phosphocellulose paper. Sometimes you'll see it called like P81 in like the old papers. So P81 is was the brand name of the original, like, or I don't know original, but the paper that was like traditionally used, and then they stopped making it. So now we get like there's a lab, the Oak Hill lab. I'll give you the link where they actually make like a substitute paper that you can use. And what's great about this paper is it's like negatively charged, and so it won't bind the ATP, but it will bind your substrate, and so your protein gets stuck on there. And now you're going to wash it. So you're going to wash off that ATP. So you do like these washes in phosphoric acid, which helps the ATP really not want to stick, and it gets breaks it and stuff, and so the ATP doesn't stick, and you get everything washed off. And you're going to dry it, and so I do like an acetone wash to help like dry it, and then acetone wash to get like rid of the acid, and then like to help with the air drying afterwards. Then I put it in these scintillation vials, so these little tubes that have this liquid in them called scintillant. And so what this is into here is a solvent, so something that solubilizes the substrate, so it's gonna like dissolve the stuff you put in there. So it's in the liquid, and then it has like a scintillant or fluorophore to capture the radiation and give it off as light. So basically, fluorescence. I've talked about this before. It's like where a molecule absorbs light at one wavelength and gives it off at another wavelength. And so basically with the liquids scintillation counting, you have the radiation being transferred to this fluorophore, so a molecule that can fluoresce, and so then this radiation is being used to excite that fluorophore and give off this light that can be detected by the machine. And I'm not going to go too much into the electronic stuff, but I'll give you a link to learn more. So then we measure that light using this really old machine that is very nostalgic to a lot of people, and everyone seems to have the same one. But you put these vials in these little racks, and then it goes through and it like moves the rack, and it counts the radioactivity that's going off, and then you also measure the radioactivity of just like your ATP mix, and then you can, like the hot ATP, so then you can calculate like the activity and the stoichiometry and all of that fancy stuff. Okay, so that's the overview, and so now some more details. So I want to start by directing you to the Newton lab. So Alexandra Newton, president of Value BMB, I've told you all about her. So she actually helped teach me how to do these, like showed me how to set up the papers really well and that sort of thing. And she has this great protocol on her lab website, freely available for anyone to go look at and read. And I'm sure if you have specific questions, she'll be happy to answer, because she really loves talking about these assays. She's a really big fan, and she's really great. So shout out to her. So this is just an example. So this is just based on what I use for my experiments. So some of it was based on the literature, so like what was already known about these kinases and these experiments. Some of it was based on like optimizing. So whenever you start like working with a new protein or a new experiment or whatever, you're often going to want to optimize for your specific one. So in this case, I'm working with the whole protein, and then I'll go into more detail on these individual components. But I mix the protein with this like ATP master mix, which has the ATP and the buffer, so like the pH stabilizing and all the salts and all that stuff, and then a kinase, and then that's like I let that reaction go, then I stop it, and then I spot wash, dry, and measure. So that's basically what we just went over, but with a little more of like concentrations and stuff. And so now let's look step by step in a little more detail. And so back to the substrate. So kinases don't just like phosphorylate anything. So we talked a little bit about how they often have like consensus sequences, but even within that consensus sequence, they only can phosphorylate specific residues. So like amino acids. So we call amino acids residues once amino acids join together to like link up to form proteins. Then we call like the part that's left over, like actually the part that's in the chain is now a residue, because it doesn't have those like amino and acid groups, because it uses them to join up. So that always confused me, and so I always probably like over explain it, and you probably didn't care, but yeah, so residue was like an amino acid, except it's like part of the chain. So anyway, different kinases can like specialize in different amino acids that they're adding to, but there's only like a set number that they can be. So it's like tyrosine, serine, and threonine are the main ones, so they could probably be like histidine in some cases, especially in like bacteria. But anyway, what these three have in common is that they have this OH group, so this hydroxyl group that the that's going to get phosphorylated. And so often kinases will be like a serine threonine kinase, so they'll do serine or threonine, or they'll be like a tyrosine kinase, so they'll do tyrosine. So you can see like serine and threonine are pretty similar, and they're both kind of smallish. But tyrosine's all big and bulky, so that's why like it's often you won't have like a kinase that's a tyrosine serine, because they're just like so different size. So the kinase is active site, so the part where it actually does the phosphorylating stuff, it's got to be like different shaped and size and stuff to actually make that happen. But serine and threonine, they can often do both. And so you can use like just the peptide or the protein. So, but when you have a peptide, you might have to alter it a little. So you might have to add basic residues to the end of the protein to get it to stick. And so by basic residues, we're talking here about like lysine and arginine, which are going to be positively charged because they've acted as bases and stolen proteins, and so they're fully proteinated, and so they're so they're positively charged. And this is important because I already went over that, but that's important because the paper that we're going to use is negatively charged, and so this is going to help it stick. And so now let's talk about that reaction buffer. So what's in here? Okay, so I call this like a master mix because I make it and then I make the same thing and then I add it to the different like sample things that I'm trying to test. So whenever you make a master mix, like make more than you need for the number you need, so make like extra because you're going to lose some and like transfer and that sort of thing. But so the main thing in here, well, I guess there's not really a main thing because they're all important, but is the ATP. So you want it to be hot. So hot we mean is like a slang for radioactivity. So we can say like something's radioactive, it's hot, and if it's not radioactive, it's cold. So the hot ATP would be like the radio labeled stuff, and then the cold ATP would be just like the normal. And you can see that. So you want it to be hot, but not too hot. You're just gonna like make your scintillator go crazy, and it's not safe, and all sorts of stuff. So I only use trace amounts of radio-labeled ATP compared to the non-radio-labeled. So in this experiment, I'm using like 100,000 times as much cold as hot. And so you can see like nanomolar, so that's like a millionth of a molar, and then micromolar, or no, nanomolar is like a billionth of a molar, and then micromolar is like a millionth of a molar. And so I'm using, yeah. So basically I have a lot more cold than I have hot. And so that is, yeah, just something to keep in mind, is that like not everything is radio labeled, because that would be like crazy hot. But yeah. And so then what else is in this buffer? So the main thing is like the buffer. Well, the first thing is like the buffer, so like the pH stabilizer. So each this might have to be optimized for your kinase. Magnesium chloride, so kinases typically use magnesium to help them do that transfer step, so you need to make sure they have plenty of that. EGTA, so you want to, this helps, this is a calcium chelator, so it like chelates, so like grabs onto and hides calcium. So you might have heard of EDTA. So EDTA will actually do that for magnesium and calcium, but it's like it will also do it for magnesium, and so that wouldn't be good because it would take the magnesium from our kinase. But the reason why you still want some sort of chelator is because it's going to help inhibit some like nucleases, so RNA and DNA chewers, and some like phosphatases, so things that will remove the phosphate, which isn't good, and it also like helps to like preserve the solution and prevent things from growing and all sorts of stuff. So good to have a chelator, but you don't want a chelator that's going to prevent your kinase from working. And so now you have, whoops, I went the wrong direction, sorry. So now we have phosphatase inhibitors. So phosphatases, remember, the things that remove the phosphorylation. So if you're trying to measure the phosphorylation, you don't want it to be removed like right after you add it. So you want to inhibit those phosphatases. And then DTT is to maintain a reducing environment. So like reducing is the opposite of like an oxidative environment or whatever. So that'll keep all like the cysteines not cross-linked and all sorts of stuff, and make it more like cell-like conditions. Okay, so now the time. It's depending on your goal. So this was on the bottom. I'm just showing you like a time course experiment where I was testing to see when I would reach like the saturated phase. So basically so I'm showing you like time and then the like moles of phosphorylation. It doesn't matter, just like you can see that it goes up over time. And so in the beginning you have this linear phase. It's hard to tell because I didn't take a bunch of time points in the beginning because I was interested more in seeing like when saturation occurred. But in the beginning you have this linear phase. And so this is when like the kinase, it has plenty of like substrate. So as soon as it finishes like phosphorylating one, it'll get another and another and another. And so you don't have to worry about it getting like depleted. So in this early time point, if you were to measure here, now this is where you want to look if you're measuring kinase activity. So like how well it works, how well it likes that substrate there, that sort of thing. And then if you want to study like, so you can do like time points or just do like an early point if you're trying to compare between things. And then if the saturation phase is like if you're doing an endpoint for stoichiometry, so like how many phosphorylations per substrate molecule. So if you were to think about this, if you had two substrates and they both had a single phosphorylation site, and you let them run to their saturation, and so like all the phosphorylations were possible sites were phosphorylated, then you would get the same number at the end if you were just to measure that endpoint. But one of the substrates might be like way worse for the kinase, so the kinase might be like way slower for one. in that initial phase, but then they both reach saturation. So if you do it like after, one might saturate like way before the other, but if you're just measuring like an endpoint at the very end, they might look the same. Whereas if you measured in the linear phase, so early on, you'll be able to tell that one was like a lot better or worse. And this is also you want to look at like you're looking at inhibitors and that sort of thing. Okay, so now the stop, the quenching part. So I have this like a master mix for this too. So I have EDTA. So now remember, EDTA was what we were avoiding before, but now we're adding it because we want to steal the magnesium from the kinase. And then we have like a huge excess of cold ATP. So before we were talking about like nanomolar, now we're talking millimolar. So like 1,000 times more than that. And this is cold, so it's not radiolabeled. And this way, if the kinase keeps going, even though we have that EDTA, anything it adds is way more likely to be like unlabeled than labeled, and so we won't detect it. It'll just be, it won't affect anything. Okay, now the P81. So this is what we talked about before, this paper. And so this is how Dr. Newton showed me how to set these up, and so I use, you can use like pins or whatever, but I didn't have pins, so I use these, what do you call those? The syringe needles. And carefully stick the papers, and the top, you want to make sure that the papers aren't touching the like styrofoam. So I'm just using a styrofoam lid from like when we buy like cuvettes, they come with like these styrofoam things with the styrofoam lid, and so I just steal the lids and use it for this little board. And so you can see that I line them up like this, and I have those three that was like don't wash. Those are the ones that I spot with my ATP mix, and that's going to allow me to convert, to compare, like, so I know how much ATP corresponds to like how much signal, and then I can use that to see how, like, compare it to how much signal I get in the various samples I spot. But you want to spot a lot less of the ATP master mix because that's going to be a lot hotter. So I normally do like 2 microliters of that and then like 15 microliters of my reaction. But it's going to depend on your protein and that sort of thing. And you want to make sure you're not adding like too much that it's not going to all soak in, because that can cause problems. So yeah, and so you want to make sure also that your filter paper should be at like the top of the sticky thing and not actually touching. And I like to line them up, so I do my experiments in like like strip tubes, and so then I just line them up how the strip tubes go, and that sort of thing. And of course this will all be done like behind a plexiglass shield in the hot room with safety and scanning and all that stuff. Okay, so sorry. Now we want to, the animations are kind of crappy because I was doing this, while also doing some other stuff. But anyway, I didn't have time to check them. But anyway, so the next step is washing it. So now we want to, so we've gotten, remember, the paper is going to stick it to get our protein stuck to the paper. And now we want to wash. And so for this, I do like 3 5-minute washes in dilute phosphoric acid. And so basically with the phosphocellulose paper, it's negatively charged, so we want it to be positively charged. And at a low pH, our substrate is going to be maximally positively charged. And so remember, we added, like, sometimes, so that's why you like add those basic peptides onto the end. And so that if we have this acid, it'll help make sure that there are plenty of protons around so that those are fully charged. But the phosphate groups, they have a really, they're like really don't want the proton. So you'd have to go like crazy low to get those not negative. So those will stay negative, but your protein or your substrate or your peptide, whatever, it's going to probably be positive. And this will allow it to stick. So yeah, before I said more on this later, now I said more on this now. But I changed the slides before, so you didn't see the more on this later. But anyway, yeah, so going back to like acid versus base, so at a low pH, you have a lot of extra protons, and so things are more likely to be protonated. Okay, so, and that makes your protein more like positively charged so you can stick. And so this idea of like getting it to stick, so this phosphocellulose is actually described as a cation exchange paper.
So this might sound familiar from when we talked about protein chromatography. We use like ion exchange chromatography. So in chromatography, we use like these little columns filled with these little beads called resin that have these different properties. And you flow these solutions containing proteins through them, and then the proteins interact differently with the resin based on the protein's properties and the resin's properties. And so then they come out at different points and that sort of thing. And so with ion exchange chromatography, we have charged resin, and then the protein binds it based on charge, and then we can heat it off. And so ions are just a word for charged particles. So basically they have an uneven number of protons and electrons. So before with the radioactivity, we're talking about protons and neutrons. Now we're talking about protons and electrons. And so neutrons are neutral, so they didn't affect the charge, but electrons do. And so if you have an imbalance of electrons and protons, you'll get an ion. If you have fewer electrons than protons, you have a cation, which is positively charged, and an anion is when you have more electrons, and so these are negatively charged. And so the idea, so with protein chromatography, you're binding this, with cation exchange, you have a positive cationic protein and a negative resin, and then you're competing it off with salt afterwards. So you get your protein to stick, and then you compete it off. But here, we're getting our protein to stick to this paper, so the paper is like the resin, but we're not trying to get it to unstick, but we just want to get the ATP and the free phosphate to not stick. So if you have that, remember we have all that extra hot ATP that doesn't get added, and so we don't want that to stick. So the point of the like paper and washers and stuff is that we're getting the protein stuck, and then we're washing off the free ATP. So remember that phosphocellulose paper, even at the low pH, is really negatively charged, and the phosphate groups are really negatively charged. So they shouldn't bind, and then the acid can also break the phosphate group off, and so then it won't bind. So you have this low pH where you have your protein or your substrate maximally positively charged, and then the ATP is still going to be negatively charged, but so is the paper, so it won't stick. So after you do those acid washes, so basically you stick the papers in this little wire mesh in this beaker, and so before you had the phosphoric acid, and so you put it in the phosphoric acid waste container. Remember, this is radioactivity, so the radioactive, and so it needs to go into the radioactive waste, and this all needs to be done behind plexiglass with safety precautions and everything. And now, after you do the phosphoric acid washes, now you're going to transfer to an acetone wash. So you're going to do the same thing, but now your beaker's filled with acetone. So like I have two beakers that I use, one for the phosphoric acid and one for the acetone, and I have two liquid waste disposals, one for the phosphoric acid and one for the acetone. And I just have it on a little stir plate. And this time I just do one acetone wash, and I'm getting rid of the phosphoric acid, and since it's acetone, it's going to help it with the air drying. And so if you think about like if you get like rubbing alcohol, how it like evaporates quickly, and so then like your skin gets all cold and stuff because the heat's getting stolen and your skin dries out. So we're like drying out the paper quicker, so it dries pretty quickly. Now we're going to add it to vials with liquid scintillant. So I typically, during the preparation stage, I put like 2 mils of the scintillant into each vial. So remember that scintillant has like the floor, it has the scintillant, and then the thing that's going to like capture the like radioactive stuff and then like pass it to the fluorophore, which is going to give it off as light. And then get measured. So I use this like MP thing. They have more details on the various formulations that they have. But basically, production of light is in this cocktail. The cocktail contains a solvent and a chemical compound known as a fluor. So they often have these benzenes as solvents. And then the fluors produce a flash of light when they interact with a beta particle. So the beta particle is like the radiation given off by, it's the type of radiation that's given off by the phosphorus. And then the photons react to the photocathode, and then that signal gets amplified, which is converted to counts per minute. So I would say that PerkinElmer has a lot of great tools about this, and I don't know what happened to my PerkinElmer tab, but I'll send the link. So we talked about it before. Okay, so now we stick it in this machine, so this scintillation counter. So now I stick in this machine, the scintillation counter, so it has these racks, and you put the vials in there, and then it takes the racks and it like moves them back and then to the side and goes one by one, and then it moves it forward and it makes these loud noises. And it's really cool. It has these little like cards, instead of like, you put these little cards on the back and it like identifies, like this little Hulk card on that red one at the end or whatever. So yeah, basically it's really old school. And it's so old school, it like prints off on like a dot matrix printer, and the computer won't save anything, so the only readout you have is that printer. So you're like deathly afraid the printer's gonna like die. But anyway, so you get this long thing of results, and then you can calculate based on like the ATP signal of just that like ATP master mix you spotted, based on, and how much of your substrate you spotted, and how much the concentration of your substrate and all sorts of things. So you have to do some calculations and stuff. But then you could work out like stoichiometry if you're doing an endpoint, so like how many phosphorylation sites per molecule, or the activity, so like how like fast and well or whatever is the kinase actually working on that substrate, and like if you did an inhibitor, did the inhibitor inhibit it, whatever. So this method's really old school, but it can be really fun, and it's really it's considered like the gold standard kind of for kinase assays. So there's a lot of like non-radioactive kinase assays now, especially, and they typically rely on measuring like ATP or ADP instead of the phosphorylated thing. So they have like more limitations and are less they can like less sensitive, I guess, and because radioactivity is super sensitive, but these other indirect methods are more susceptible to like influence from non-substrate things, and so there's various reasons why this method is preferred, but it's also more like tedious and time consuming, and harder to do with like high throughput, so if you have a ton of samples. So I think I've done like 100 or so at a time, but yeah. And yeah, one of my favorite things about this machine is that there's this glitch in the software, so it can't go past like 2000, so it thinks it's 1921. So yeah, so I hope that helps you understand these experiments, and thanks again to Dr. Newton for helping me understand these experiments better, and set them up maximally efficiency, whatever. And also thanks to Nicholas Tonks for letting me use his machine, and for helping teach me, as well as a postdoc in his lab, Prabha, who taught me directly and is really great. So thanks all, and hope that helped.